வாகை சூடவா.
Dear students, I like to give a brief introduction about genetics. You know genetics is nothing but the study of heredity and inherited variation. You know what is heredity and what is variation. Before going to the heredity and variation, the term genetics coined by the scientist W. Batson in the year 1906. Coined or introduced, it's very important for your NEET examination. Heredity means the transformation of characters from parents to the subsequent generation. Transformation of characters from parents to the subsequent generations, or transformation of characters from the parents to the offspring is also said to be heredity. Then variation means the offsprings differ from their parents in slight degree is said to be variation. So study of heredity and inherited variation is said to be genetics, and already you know the father of genetics, Gregor Johann Mendel. You know variation. Variation is nothing but the offsprings differ from their parents either in slight degree or they show large variations. Mutation and crossing over mainly responsible for variation in organisms. You know mutation. Mutation is nothing but sudden spontaneous deviation from the wild type. Then variation, mainly crossing over, plays a vital role. What is crossing over? The exchange of genetic materials takes place between the non-sister chromatids of the homologous chromosome is said to be crossing over. So variation due to either mutation or crossing over. And two types of variation generally reported in plants as well as animals or in organism. One is called discontinuous variation, another is called continuous variation. If the variation inherited to the subsequent generations, we are calling it as continuous. If it is not continuous one, it is not inherited to the subsequent generations, we are calling it as discontinuous one. One is heritable. Heritable variations are known as continuous variations. Another is non-heritable. The non-heritable variations are known as the discontinuous variations. Then the importance of variation, these things are very important for your NEET examination. Variation makes some individuals better fitted in the struggle for existence. According to Darwin, natural selection, organisms show variation is the fittest organism selected by the nature. Like that, if the organism or if the plant or the animal show variation, it is better fitted in the struggle for existence, and they help the individuals to adapt themselves to the changing environment. Nothing permanent in the world except change. So we should face the changes, we should adapt to the changes of the environment. It provides a genetic material for natural selection, because at the time of variation we show differences from other organisms due to crossing over or mutations, and the variant material naturally selected by the nature to the subsequent generations, and finally they constitute the raw material for evolution. Crossing over and mutation responsible for recombination, it leads to the formation of new varieties and plays an important role in evolution. So variation due to mutation as well as crossing over plays a vital role in the evolution process. So it is a raw material for evolution. Mendelism, you know, Gregor Johann Mendel, father of genetics. In the NEET examination, the birthplace of Gregor Johann Mendel is vital. Birthplace of Gregor Johann Mendel is Heinzendorf in Czechoslovakia. Then he did monohybrid cross, monohybrid back cross, monohybrid test cross, dihybrid cross, dihybrid back cross, dihybrid test cross, and formulated 4 laws. One is law of unit character, another is law of dominance, third is law of independent assortment, fourth is law of purity of gametes in his book Experiments in Plant Hybridization. And Gregor Johann Mendel described and catalogued around 24,034 plants in Pisum sativum. So Mendel's experiment material, you know, Pisum sativum, the common name of Pisum sativum is garden pea plant. So book by Gregor Johann Mendel is Experiments in Plant Hybridization. It's an important one. for your NEET examination. Mendel formulated monohybrid, monohybrid back cross, monohybrid test cross, dihybrid back cross, dihybrid test cross. And how he done the experiment? The cross pollination in Pisum sativum. Pisum sativum with bisexual flower. Bisexual flower, androecium and gynoecium present in the same flower, we are calling it as bisexual. Mendel selected one plant as male and another plant as female. He transferred pollen grain from the male plant to the stigma of the female, where cross pollination takes place. Male flower, the plant possesses bisexual flower. He considered one plant as male. From the male plant, he took the pollen grain and transferred into another plant. In the another plant behaved like a female, so he removed the anther or stamen from the bisexual flower. The process is said to be emasculation, removal of anther or stamen from the bisexual flower and converting the bisexual flower into the female flower is said to be emasculation. So in this way he crossed tall with the dwarf, yellow with the green, round with the wrinkled, like that. Then we come to the characters selected by Mendel for his experiment. Actually, Mendel found around 34 varieties in the garden and he selected 7 varieties for his experiment, and the characters selected by Mendel for his monohybrid and dihybrid cross are: character number 1, stem length, tall is dominant over dwarf; second is pot shape, inflated dominant over constricted; seed shape, round is dominant over wrinkled; and seed color, yellow color is dominant over green; and flower position, axillary flower is dominant over the terminal flower; and flower color, purple color is dominant over white, and pot color, green, is dominant over yellow. These things are very important for your NEET examination. So tall is dominant over dwarf, inflated is dominant over constricted, round is dominant over wrinkled, yellow is dominant over green, axillary flower is dominant over terminal, purple flower is dominant over white, and green part shape is dominant over yellow. These things are very important for your NEET examination. Then Mendel selected 7 characters for his experiment. Pisum sativum plant with 14 chromosomes. Technically says it is present in 7 pairs, 7 homologous pairs. These things are also important for your NEET examination. The number of chromosomes present in Pisum sativum is 14. That means 7 pairs of homologous chromosomes are present. The character determine flower color and seed color located in the chromosome number 1. The character determined the character part shape and flower position determined by some factors, because Mendel called genes as factors. The factor responsible for part shape and flower position located in the chromosome number 4, and the genes responsible for controlling plant height is also present in the chromosome number 4. The character, the genes or the factor controlling the pot color located on the chromosome number 5, and the factor which control seed shape located on the chromosome number 7. So location of the genes or factors and the chromosome number is very important for your NEET examination. Flower color and seed color on the chromosome number 1, and the flower position, pot shape and plant height located on the chromosome number 4, pot color located on the chromosome number 5, and seed shape, the location is chromosome number 7. These things are very important for your examination. Then we come to what are the important terminologies you should have a clear idea for Mendelian experiment or basic genetics. Number 1 is gene. Mendel called genes as factor. Number 1, the gene or factor. Number 2, allele. Number 3, allelomorph. Number 4, F1 generation or first filial generation. Number 5, F2 generation, the second filial generation. Then phenotype, then genotype, then dominant character, then recessive character. These things are very important for, these terminologies are very important for your NEET examination. Gene or factor, the basic hereditary unit is said to be gene or factor. Mendel called genes as factor. Example, capital T gene is responsible for tallness and small t gene is responsible for dwarfness. Define an allele, altering forms of the same gene which controls different expressions of a single character. For example, height is the character. More height, 180 centimeter tall plant, less 45 centimeter dwarf plant. According to Mendel, the factors are expressed in two ways. One is called dominant, another is called recessive. Define an allele, altering forms of the same gene which controls different expressions of a single character. Here the character is one, height. It is expressed in two ways. One is called tall, another is called the dwarf. So the genes are factor controlling tallness and dwarfness, collectively known as an allele, or altering forms of the same gene which controls different expression of a single character is said to be an allele. The character produced by the allele is said to be allelomorph. The position of a gene or an allele in a chromosome is said to be locus. Then F1 generation, the generation produced by crossing two pure parental stocks. What is F2 generation? The generation produced by crossing two F1 hybrids. Then what is a phenotype? Physical expression of a character is said to be phenotype. What is genotype? Genetic expression of a character is said to be genotype. What is a dominant character? According to Mendel, whenever a cross made between two pure parental stocks, the F1 hybrid produces any one of the parental character. The character expressed in the F1 generation is said to be the dominant character. The character which is not expressed or suppressed in the F1 generation is said to be the recessive character. And two things you should remember. One is homozygous condition, another is heterozygous condition. Homozygous means the organism with two similar genes or two identical genes or two identical factor is said to be homozygous. Example, capital T responsible for tallness, or small t responsible for dwarfness. Define heterozygous condition, an organism with two different genes or dissimilar genes is said to be heterozygous condition. Example, capital T, small t. These things you should remember for all Mendelian experiment, intergeneric, intrageneric experiment. These things you should remember. So gene, allele, locus, allelomorph, homozygous, heterozygous, phenotype, genotype, dominant and recessive. Next is Mendelian inheritance after monohybrid and dihybrid cross. Mendel formulated law of dominance, law of segregation, law of purity of gametes, and in the end of dihybrid cross he formulated law of independent assortment. Three laws in the end of monohybrid cross: law of dominance and law of purity of gametes or law of segregation. In the end of dihybrid cross, Mendel formulated law of independent assortment. Then monohybrid cross. To read this monohybrid cross in the 10th standard itself, anyhow I like to give a brief introduction about the monohybrid cross. A cross made to study the inheritance of single pair of related character is said to be monohybrid cross. single pair of related character means according to Mendel, the character is always expressed in two ways. One is called the dominant character, another is called the recessive character. So definition, a cross made to study the inheritance of single pair of related character. Experiment conducted by Gregor Johann Mendel. Experiment material, Pisum sativum. The common name is garden pea plant, important for your NEET. Character selected for the experiment. For monohybrid cross, Mendel selected a height of the plant or height of the stem. Stem, the perfect is height of the plant. Tallest dominant over dwarf. Mendel crossed a pure homozygous tall plant with a pure homozygous dwarf plant. Homozygous means the organism with a similar genes or identical genes. Mendel crossed a pure homozygous tall plant with a pure homozygous dwarf plant. The F1 generation produced all heterozygous tall plants. Heterozygous means the organism with two dissimilar genes or two different genes. F1 generation produced all heterozygous tall plants. So tall is the dominant character, dwarf is the recessive character. According to Mendel, whenever a cross made between two pure parental stocks, the F1 hybrid produces any one of the parental character. The parental character expressed in the F1 generation is said to be the dominant character. The character which is not expressed or suppressed in the F1 generation is said to be recessive character. On selfing, the F2 generation produced 3:1 ratio of tall is to dwarf. This is said to be the phenotypic ratio. The two different genes present in the F1 hybrid separated at the time of gamete formation. This is called law of segregation or law of purity of gametes. Mendel crossed tall with dwarf. F1 generation produced all heterozygous tall. On selfing, the F2 generation produced 3:1 ratio of tall is to dwarf. This is said to be the phenotypic ratio, physical expression of a character. Then Punnett formulated this Punnett square. This is male F1, this is female S1, female F1, capital T small t, you know the cross, capital T, capital T, capital T small t, capital T small t, small t is here. The genotypic ratio, the phenotypic ratio 3:1. See here, the genotypic ratio is 1 homozygous tall, 2 heterozygous tall and 1 homozygous dwarf. So the genotypic ratio of this cross is 1:2:1. These two are important for your NEET examination. So the phenotypic ratio of monohybrid cross is 3:1. The genotypic ratio of monohybrid cross is 1:2:1. This is monohybrid cross. Then you know back cross and test cross. Back cross means crossing the F1 hybrid with any one of its parent. Crossing the F1 hybrid with any one of its parent is said to be back cross. What is a test cross? Crossing the F1 hybrid with its recessive parent is said to be test cross. F1 hybrid, you know, Mendel cross a pure homozygous tall with a pure homozygous dwarf. F1 generation produced all heterozygous tall. So tall is the dominant character and dwarf is the recessive character. What is test cross? Crossing the F1 hybrid with its recessive parent is said to be test cross. It results in 1:1 ratio of tall is to dwarf. This is monohybrid test cross ratio. Here you get two gametes, capital T, small t. Here similar gametes, small t, small t. If you cross this capital T with small t, you get capital T, small t. If you cross small t with small t, small t, small t, here it is 1:1. So monohybrid cross ratio, phenotype is 3:1, genotype is 1:2:1. Monohybrid test cross ratio is 1:1. This ratio is also important for your examination. Then back cross, already I informed what is back cross. Then dihybrid cross. What is dihybrid cross? A cross made to study the inheritance of two pairs of contrasting character is said to be dihybrid cross. A cross made to study the inheritance of two pairs of contrasting character is said to be dihybrid cross. Experiment conducted by Gregor Johann Mendel, experiment material, Pisum sativum, character selected for the experiment, cotyledon color and seed form or seed color and seed form. Yellow color is dominant over green. In the cotyledon, yellow is dominant over green. In the seed form, round seeds are dominant over wrinkled. Then you know the explanation. We need only the ratio for the NEET examination. Mendel crossed a pure homozygous yellow round seed giving pea plant or round yellow seed giving pea plant with green wrinkled. The F1 generation produced all heterozygous yellow round seed giving plants. Mendel crossed a pure homozygous yellow round seed giving pea plant with a pure homozygous green wrinkled seed giving pea plant. The F1 generation produced all heterozygous yellow round seed giving plants. You know, heterozygous means dissimilar genes, homozygous means similar genes or similar factors. On selfing, the F2 generation produced 9:3:3:1 ratio of yellow round, yellow wrinkled, green round and green wrinkled seed giving plants. Mendel crossed a pure homozygous yellow round with the green wrinkled. F1 generation produced all heterozygous yellow round seed giving plants. So, yellow round is the dominant character, green wrinkled is the recessive character. On selfing, the F2 generation produced 9:3:3:1 ratio of yellow round, yellow wrinkled, green round and green wrinkled seed giving plants. Yellow round and green wrinkled are the parents. Yellow wrinkled and green round are the recombinants or new varieties are formed in the end of dihybrid cross. How these recombinants are formed? For the formation of this recombinant, Mendel formulated law of independent assortment. F1, see here, the F1 hybrid with 4 different factors, 2 from the paternal side and 2 from the maternal side. Capital Y, capital R from the paternal side, small y, small r from the maternal side, they get separated or assorted independently at the time of gamete formation. It is said to be law of independent assortment. See here, recombinants here, yellow wrinkled and green round or the recombinants or new varieties are formed in the end of dihybrid cross. This is important for your NEET examination. So, how these yellow wrinkled and green round seeds are formed? It is due to law of independent assortment, recombinance, NANTS, recombinance. How these recombinants are formed? It is due to law of independent assortment. See the F1 hybrid with 4 genes or 4 factor, 2 from the paternal side, capital Y, capital R from the paternal side and small y, small r from the maternal side. They get assorted independently at the time of gamete formation. Due to independent assortment, 2 new gametes are formed, namely capital Y, capital R and small y, small r. This new gametes responsible for the formation of new varieties. So, this ratio is said to be dihybrid cross ratio, dihybrid phenotypic ratio. The genotypic ratio of dihybrid cross is 1:2:1, 2:4:2 and 1:2:1. Monohybrid phenotypic ratio 3:1. Monohybrid genotypic ratio 1:2:1. Monohybrid test cross ratio 1:1. Dihybrid cross phenotypic ratio 9:3:3:1. Dihybrid cross genotypic ratio 1:2:1, 2:4:2 and 1:2:1. The dihybrid test cross ratio, test cross means crossing the F1 hybrid with its recessive parent. So you should remember dihybrid test cross ratio is 1:1:1:1. In NEET examination they ask pick out the correct pair or pick out the incorrect pair like that, or pick out the correct answer or pick out the incorrect answer like that. They ask these kind of questions. These ratios are very important for your NEET examination. Dear students, now I am going to discuss important questions in genetics, the advancement in genetics, the gene interaction or factor interaction, the mutation, ploidy, blood group, as well as the various syndromes, Klinefelter syndrome, Turner syndrome, Down syndrome, like that. The question number 1 for you: genes for cytoplasmic male sterility in plants are generally located in. You know sterility, unable to produce fertile offspring, it is said to be sterile. In plants, sterile stamen is said to be staminode, and you know sterile carpel is said to be pistillode. Staminate means the male flower, pistillate means the female flower, staminode means the sterile stamen, and pistillode means the sterile carpels. You have clear idea about this. So the question is, genes for cytoplasmic male sterility in plants are generally located in. Option A, mitochondrial genome. Option B, the cytosol. Option C, the chloroplast genome. The option D, the nuclear genome. Before going to the answer, you should know what is genome. I hope you have clear idea regarding the genome. Genome means DNA that resides in a cell is said to be genome, or haploid set of chromosomes present in a species is said to be genome, or DNA that resides in a cell, either a somatic cell or the gametic cell, plus the cell organelles present in the cell, or the DNA present in the cell organelle like mitochondria and chloroplastid, collectively known as genome. Simply says, genome means DNA content of a cell. So DNA content of a cell is said to be genome. Generally, characters in organisms controlled by the chromatin reticulum present inside the nucleus. This is nucleus, controlling center of a cell, with nuclear membrane, nucleopore, nucleoplasm and the nucleolus, and it possesses the chromatin reticulum. Chromatin reticulum is nothing but the DNA plus histones. The genetic material or the genes, the DNA or the nucleotides present in the chromatin reticulum present inside the nucleus controls the characters. At the time of cell division, this chromatin reticulum condensed to form the chromosomes, you know pretty well. The question is, genes for cytoplasmic male sterility. What is the role of cytoplasm? In certain cases, the genes present in the cytoplasm other than the nucleus may control the characters, may control the expressions. For example, in the previous classes you read about the semi-autonomic bodies. Mitochondria and chloroplastids are very good examples for semi-autonomic bodies. Semi-autonomic bodies means mitochondria as well as chloroplastid possesses circular DNA plus 70 as ribosomes. With the help of this circular DNA, mitochondria as well as chloroplastids control itself. With the help of ribosomes, these mitochondria as well as chloroplastid produce their own proteins, protein synthesis. So these two are known as the semi-autonomic bodies. If the genes or if the DNA present in these semi-autonomic bodies either in mitochondria or chloroplastid control a particular trait, control a particular character. We are calling it as cytoplasmic inheritance. If the DNA present in the cytoplasm or DNA present in the mitochondria or DNA present in the chloroplastid control a character, control inheritance, we are calling it as cytoplasmic inheritance. Here the question is cytoplasmic male sterility controlled by, or the genes responsible for cytoplasmic male sterility located in. The answer is option A, mitochondrial genome. That means the genes or the DNA present in the mitochondria controls male sterility. So this is the answer for you. So correct option for this question is, correct answer for this question is option A, mitochondrial genome. So simple definition, if the genes present in the cytoplasm or DNA present in the cytoplasm or DNA present in the mitochondria or DNA present in the chloroplastid controls the body character or controls the character of an organism, it is said to be cytoplasmic inheritance. The next question for you, in which mode of inheritance do you expect more maternal influence among the offspring? You know, we receive genes from the paternal as well as maternal side. If we receive the chromosome from the father, we are calling it as paternal. From the mother, we are calling it as maternal. We have 46 chromosomes, 23 chromosomes from the paternal side and the 23 chromosomes from the maternal side. That's why we have 46. The question is, in which mode of inheritance do you expect more maternal influence among the offspring? One is autosomal. You know what are autosomes? The body chromosomes are otherwise known as autosomes. Second is cytoplasmic, the DNA present in the mitochondria or DNA present in the chloroplastid. Number 3, Y-linked. If the genes present in the Y chromosome control the body character, we are calling it as Y-linked genes. And next is X-linked. If the genes present in the X chromosome control the body character, we are calling it as X-linked. Read the question once again. In which mode of inheritance do you expect more maternal influence among the offspring? The correct answer for the question is B, cytoplasmic. Question, read the question. In which mode of inheritance do you expect more maternal influence? Maternal, actually, we receive the cytoplasm only from the mother at the time of gametic fusion. Fusion of male and the female gametes, and it results in the formation of a diploid cell called zygote. In this fertilization, the male supplies only the nucleus, the female supplies nucleus as well as the cytoplasm. So maternal influence is more in cytoplasmic inheritance. And the next question, a pleiotropic gene. What is pleiotropic? One gene controls more than one expression. Actually, according to Mendel, a gene or a pair of gene controls a phenotype. More than a pair of gene or more than a pair of alleles controls phenotype, then Mendelian ratio gets modified. And according to Mendel, the characters are always expressed in two ways. One is called the dominant, another is called recessive, and it is controlled by two alleles, capital T and small t. One character with more than two expression is said to be multiple allelism or polymorphism, and it is controlled by a pleiotropic gene. See the option A controls multiple traits in an individual. Option B, it expressed only in primitive plants. Option 3 is a gene evolved during Pliocene, and option D controls a trait only in combination with another gene. The correct answer for the question is answer A, controls multiple traits in an individual. So in a particular person, many characters controlled by a single gene. The gene is said to be pleiotropic gene. And the next question, translocation mutation takes place between. What is translocation? First, you know what is translocation. Translocation is a kind of chromosomal aberration or chromosomal abnormality or chromosomal mutation. What is chromosomal aberration or chromosomal abnormality or chromosomal mutation? Sudden change takes place either in the structure of chromosome or number of chromosome, we are calling it as chromosomal aberration or chromosomal abnormality or chromosomal mutation. The structural chromosomal mutation, the chromosomal aberration, change in the structure of a chromosome is of 4 types, namely deletion or deficiency, duplication, inversion and translocation. So, translocation is a kind of chromosomal aberration or chromosomal abnormality or chromosomal mutation, mainly responsible for species differentiation and plays a vital role in evolution of species. What is translocation? Exchange of genetic material takes place between the non-sister chromatids of the homologous chromosome is said to be crossing over. Exchange of genetic materials takes place between the non-sister chromatids of the homologous chromosome is said to be crossing over. Translocation means exchange of genetic material takes place between non-homologous chromosome, so it is said to be illegitimate crossing over. So, the correct answer, the option A, non-sister chromatid, option B, sister chromatid, option C, homologous chromosome, option D, non-homologous. So, translocation mutation is nothing but exchange of genetic material takes place between morphologically dissimilar chromosome. The morphologically dissimilar chromosomes are known as non-homologous chromosome. The correct option for the question number 4 is option D. This translocation mutation takes place between non-homologous chromosome. So, it is also known as illegitimate crossing over. This translocation mutation responsible for species differentiation and plays a vital role in origin of new species. And the 5th question for you, in a mutational event, when adenine is replaced by guanine, it is a case of frameshift mutation, transcription, transition and transversion. You know what is mutation? Mutation means sudden spontaneous deviation from the wild type. The term mutation coined by the scientist Hugo de Vries, and Hugo de Vries first reported mutation in Oenothera lamarckiana, the evening primrose plant. Charles Darwin called mutation as sports and Batson called mutation as discontinuous change. So, mutation, sudden change. If sudden change takes place in a nucleotide or a pair, we are calling it as gene or point mutation. So, the correct answer for the 5th question is option A, frameshift. Frameshift mutation involves the changes in the nitrogenous bases. Transcription mutation, transition, transversion. Transcription, you know the process. Transcription is a process, the conversion of DNA into mRNA is said to be transcription. Transition means a purine base replaced by a purine or a pyrimidine base replaced by a pyrimidine. We are calling it as transition. Transversion means a purine. Transition means a purine base replaced by a purine or a pyrimidine base is replaced by a pyrimidine. We are calling it as transition mutation. A purine base replaced by a pyrimidine or a pyrimidine base replaced by a purine, we are calling it as a transversion mutation. See here, adenine and guanine are the purine bases, thiamine and cytosine are the pyrimidine bases. If a purine is replaced by a purine or a pyrimidine is replaced by a pyrimidine, we are calling it as transition. A purine by a pyrimidine or a pyrimidine by purine is called a transversion. The question is, in a mutational event, when adenine is replaced by guanine, the correct answer for the question number 5 is option C, transition mutation, because you know the nitrogenous bases, 2 types of nitrogenous bases reported in organism. One is the purine base, another is the pyrimidine base. Adenine and guanine are the purine bases, thiamine and cytosine are the pyrimidine bases. So, transition means adenine replaced by guanine, a purine is replaced by purine. Then the next question, mutation can be induced with. Mutation, the substances, mutation, the sudden change. Mutation is induced with the help of mutagens or the mutagenic agents. The chemical substances or the light or the temperature used to induce, used to stimulate mutation are known as mutagenic agents or mutagens. Question is, mutation can be induced with option A, gamma radiation, option B, infrared radiation, option C, IAA, and option D, ethylene. You know, ethylene is a gaseous hormone responsible for ripening of fruit. IAA, the first discovered hormone, it's a natural auxin. IAA means indole 3-acetic acid. Auxion means to grow, you know, read in the 11th standard. It's a plant hormone, it's a phytohormone. You know, hormones or the chemical substances used to control, regulate, coordinate various metabolic activities in plants. Auxins, gibberellins, cytokinins, abscisic acid, ethylene are all example for hormones. IAA is a very good example for natural auxin. So, what is the role of IAA? Auxion means to grow at low concentrations. It stimulates the growth of stem and inhibits the growth of root. So, the hormone responsible for elongation of stem and inhibits the growth of root is said to be auxin. So, IAA is a hormone, no role in mutation. Infrared radiation also not affect the genetic makeup of the organism. So, the correct answer for the question number 6 is gamma radiation causes or stimulate mutation in plants as well as animals. Then next question, question number 7. The mechanism that causes a gene to move from one linkage group to another is called. The mechanism that causes a gene to move from one linkage group to another is called. Option A, translocation. Option B, crossing over. Option C, inversion. Option D, duplication. You know what is translocation? The transfer of genetic material or exchange of genetic material takes place between non-homologous chromosomes. Second is crossing over. The exchange of genetic material takes place between the non-sister chromatids of homologous chromosome. Inversion mutation, the genes are arranged in the reverse order. Duplication, you know, the small portion of the chromosome segment represented twice. What is linkage? Here they mentioned the mechanism that causes a gene to move from one linkage group to another. Coexistence of genes in the same chromosome during inheritance is said to be linkage. Coexistence of genes in the same chromosome during inheritance is said to be linkage, and the genes are known as linked genes. Here the question is the mechanism that causes gene move from one linkage group to another linkage group. The answer is translocation mutation, because here the exchange of genetic material takes place between non-homologous chromosomes. So, this is the correct answer. The next question, a point mutation comprising substitution of a purine by a parabidin is called. Already we read about this, I explained about this mutation. Point mutation is nothing but sudden change in a nucleotide or a pair. More than a pair of nucleotides are involved, we are calling it as gross mutation. And this point mutation or gene mutation of 3 types, namely deletion, addition and substitution. Removal of a nucleotide is said to be deletion, addition of a nucleotide is said to be addition, translocation, here translocation, you know, translocation takes place between non-homologous chromosome. Third kind is substitution mutation. If a purine base is replaced by a purine or a pyrimidine base is replaced by a pyrimidine, it's called a transition. Here the question is purine is replaced by a pyrimidine. If a purine base is replaced by a pyrimidine base or a pyrimidine base is replaced by a purine base, we are calling it as a transversion mutation. So, the correct answer for the question number 8 is transversion. Transversion is a kind of gene or point mutation where exchange of nitrogenous bases or a purine is replaced by a pyrimidine base or a pyrimidine base is replaced by a purine base. The substitution mutation is genetically important because it causes phenotypic changes in organisms. Next question, question number 9. Frame shift mutation occurs when? Option A, base is substituted. Option B, base is deleted or added. Option C, anticodons are absent, none of this. If the base is substituted, a purine by purine or a pyrimidine by pyrimidine, we are calling it as a substitution mutation. It may be either transition or transversion. Base is deleted or added. Addition or deletion, deleted or added is the correct answer, because during frame shift mutation, a nitrogenous base or a nucleotide is deleted or added. Correct answer for the question 9 is option B, base is deleted or added. It may be either a deletion mutation or addition gene or point mutation. Next, the question number 10. The distance between two genes in a chromosome is measured in crossover units, which represent option A, ratio of crossing over between them, option B, percentage of crossing over between them, option C, number of crossing over between them, and option D, none of the above. The distance between two genes, actually distance between two genes. See here, this is the chromosome. This is locus 1, locus 2, locus 3, locus 4, locus 5, locus 6, locus 7, locus 8. Mentioned it as A, B, C, D, E, F, G, H, like that. If the genes are nearer, the rate of crossing over is less. If the genes are further apart, the rate of crossing over is more. So the rate of crossing over depends on the distance between the genes. Already you know pretty well the answer. So crossing over, the exchange of genetic material. If the genes are nearer, the rate of crossing over is less. They are called tightly linked genes. If the genes are further apart, the rate of crossing over is more. We are calling it as loosely linked genes. In this case, the correct answer is for the question number 10. The distance between two genes in a chromosome is measured in crossover units, which represent percentage of crossing over between them. This is the right answer for the question number 10. Then question number 11. When a cluster of genes show linkage behavior, they, option A, they do not show a chromosomal map. Option B, show recombination during meiosis. Option C, do not show independent assortment. Option D, induce cell division. So group of genes show linkage. The correct answer, question number 11, is option C, they do not show independent assortment. So linked genes do not show independent assortment if the rate of linkage is somewhat less. If they are loosely linked genes, then they show law of independent assortment, they follow law of independent assortment. The next question for you, genetic map is one that. Genetic map is one that. What is genetic map? Before going to say something about the genetic map, the options are option A, establish sites of the genes in a chromosome. Option B, establishes the various stages in gene evolution. Option C, shows the stages during cell division, and option D, shows the distribution of various species in a region. Genetic map or the chromosomal map. See the chromosome, genetic map or the chromosomal map helps to find the distance between the genes in the chromosome, the location of genes in the chromosome. See, this is a chromosome with the part centromere, and it also have chromatid. The position of a gene or an allele in a chromosome is said to be locus. See, consider this point as a gene. The position of a gene or an allele in a chromosome is said to be locus. And the genes are arranged in a linear manner on the chromosome. Genetic map helps to find out the distance between genes in the chromosome, the location of genes in the chromosome, as well as the rate of crossing over. If the genes are nearer, the rate of crossing over is less. The genes are further apart, the rate of crossing over is more. Then the answer for the question number 12 may be option A, establish sites of the genes on a chromosome. Sites represent the locus. Position of a gene or an allele in a chromosome is said to be locus. So genetic map is one that establish sites of the genes on a chromosome. So chromosome on the arrangement of genes in a chromosome, the scientists classified the genes into two types for linkage case. Linkage, coexistence of genes during inheritance is said to be linkage. The genes following are transmitted to the subsequent generation continuously are known as the linked genes. If the genes are nearer, then the rate of crossing over is less, called tightly linked genes. The genes are further apart, the rate of crossing over is more, so loosely linked genes. That means the crossing over depends on the distance between the genes. Nearer, rate of crossing over is less. If the genes are further apart, the rate of crossing over is more. This is regarding the genetic map. And the last question, after a mutation at the genetic locus of the character of an organism changes due to the change in. See the question, after a mutation, sudden change at a genetic locus of the character of an organism changes due to changes in option A, DNA replication, option B, protein synthesis pattern, option C, RNA transcription pattern, and finally protein structure. The correct answer for the question number 13 is protein structure. You know, proteins are known as the bodybuilders, chemically made up of amino acids. Amino acid combines to form a protein, amino acid, peptide bond, dipeptide bond, and polypeptide bond. So the correct answer for the question number 13 is option D, protein structure. Dear kids, these questions somewhat help you to score good marks in the NEET examination. All the best. Best wishes.
இந்நிகழ்ச்சியை பற்றிய தங்களின் மேலான கருத்துக்களை கீழ்க்கண்ட முகவரிக்கு கடிதம் மூலமாகவோ மின்னஞ்சல் மூலமாகவோ குறுஞ்செய்தி மூலமாகவோ தெரியப்படுத்தலாம். அனுப்ப வேண்டிய முகவரி சிறப்பு அலுவலர், கல்வி தொலைக்காட்சி, 8ஆம் தளம், அண்ணா நூற்றாண்டு நூலகம், காந்தி மண்டப சாலை, கோட்டூர்புரம், சென்னை 6,85,000. தொலைபேசி எண் 7824015000.